So what I'm going to do for the next 25 minutes or so is really demonstrate how if you're an inquisitive clinician with an access to some research laboratory and training in research laboratory, we could try and make a difference to patients and by trying to understand what is exactly going on to patients and improve care of patients. So it's really a translational research, and over the next 20 minutes I'll highlight what has been done in Hamilton over the past 25 years or so. So it's really to understand components of asthma. You heard from Dr. Byrne and from Ed that your airways close in. So when a tube closes, there are only two ways the tube would close. The muscles lining the tube would contract, or something fills up, goop will fill up the tube. So you heard about sputum, the old technique of rolling you over and tapping your chest to get rid of the goop. So if a tube plugs up, it's something that causes spasm, which is a variable airflow obstruction, spasm of the muscles that line the tube, or something filling up the tube. So we'll try to understand how could we distinguish in an individual patient what is contributing to the obstruction, and how do you assess what is in the goop. If you have got secretions, you've got to examine them. So in order to treat an organ, we always have to examine the secretions of that organ. It's common sense, isn't it? If you've got diarrhea, well, it's a sort of shitty thing to talk about, but you examine your feces. If you've got heart problem or blood problem, you examine your blood. If you've got a kidney problem, you examine your urine. So if you've got a lung problem, you should examine your sputum. It's a bit yucky, but you've got to do it. So we'll talk about assessing bronchitis. And we'll talk about assessing one particular blood cell in the eosinophil, the pink cell, which is what I like, the color and the biology of the cell, which we'll talk about. And then finally illustrate how, if you understand the biology and understand why a particular cell gets into the lung, we could really target that cell. And then we'll get a bit more complicated in patients with severe asthma. Why does this cell get into the lung and what can be done about it? And how could we assess if that cell is present or not by simple methods? So this is essentially what I'm going to talk about in 20 minutes. So we've already talked about this. Ed has symptoms of cough and wheeze. So what exactly is contributing to that? Is it that variable airflow obstruction, which is spasm? Or is it bronchitis, which is inflammation and cells and exudate filling up the tubes? There are only two ways that they could get blocked up. So which of the two are contributing? And once we make that measurement, it's very easy to treat patients with asthma. Every patient with asthma has got the variability. That is a defining feature of asthma. There is always spasm, but not all patients may have got cells filling. So if we can find out which patient has got what and when, the treatment becomes much better. It may not be relevant for the patient with mild asthma. You treat them with a bronchodilator that would open up the airways, and you treat with an inhaled corticosteroid, which you heard, which would clear out the inflammation. But in patients such as Ed, who has got more severe asthma, who at one point in his life required 15 to 20 milligram of prednisone, which really only affects that inflammatory component, it's important to know if there is inflammation. So how would we assess inflammation? Does anyone know who this person is? Very venerable looking, scientist, peering over his glasses, It's Paul Ehrlich. So Paul Ehrlich got the Nobel Prize in Medicine Physiology in 1908 for studies related to immunity. He made seminal observations regarding diphtheria antiserum, and he shared the Nobel Prize with Mechnikov in 1908. But he was a cell. He was born in Silesia, present-day Poland, I think, and his father, he had access to lots of dyes because his father was in the dye industry. He used aniline dyes and started staining all sorts of cells. It was around the time that Robert Koch was doing experiments with tuberculosis, trying to stain the Mycobacterium tuberculosis. And because his father was in the dye industry and he had access to dyes, he started experimenting, staining blood cells and sputum cells with dyes, and came up with the description of the eosinophil. Eos is pink. He found that these cells, one of the blood cells, stained pink, beautiful pink cell, the Barbie cell, with a lot of granules inside, and when the cell bursts, the granules come out and it causes its damage. These cells go up in sputum and in blood when you've got an allergic reaction. And this is the earliest evidence and the description of allergic cell in blood and sputum. And he described a method to stain blood and sputum for these cells. However, it was never applied in clinical practice for many, many years until this gentleman, Dr. Harry Moro Brown, restarted examining this in clinical practice. Now, Harry Moro Brown was a physician who trained in Scotland and then finished his internal medicine and respiratory medicine training and then set up practice in central England in Derby. It was around mid-50s, and the mighty Medical Research Council of the United Kingdom, basking in its glory from conducting the first streptomycin clinical trial, had just finished a second trial showing that corticosteroids are not effective in the treatment of asthma. And you know how well they were wrong. And Harry Moro Brown just could not believe that the Medical Research Council had just published in The Lancet, one of the, in those days, the most prestigious medical journal. He just didn't believe it, even though it had come from the Medical Research Council of the United Kingdom. Because he had observed that every patient of his who had what he thought was allergic asthma got better with prednisone. And every patient who did not have allergic asthma did not get prednisone. So what he did was he set up a little clinic, got a microscope, and started looking at the sputum, staining them with the same method that Paul Ehrlich had described earlier, and observed eosinophils, and he started giving steroids only to those patients who had eosinophils. But he wanted to do a randomized controlled trial. The first major randomized controlled trial ever. You're in the temple of evidence-based medicine. You have to know about randomized controlled trial. The first major trial was streptomycin for tuberculosis. So he wanted to do a randomized controlled trial, but no one would fund him because MRC had just shown that it didn't work. So he got Allen and Hanbury's, in those days Glaxo, Allen and Hanbury's, to give him some money and some medication to do a study to see if those cells, eosinophils in sputum, this was his original stain. He sent them to me just a few months before he died, and that's another story. He unfortunately passed away last year. I had written an editorial in a journal and I had forgotten to reference him. He was 96 at that time. He read that article and sent me an email chastising me for omitting his original 1958 article. So this shows the commitment of this person, a true translational scientist, to patient care and applying science to the practice of medicine. He told me that even at that age, at 96, he was practicing allergy. He had his old microscope, and he would see patients, get them to spit, stain the sputum, and give treatment, which is way better than what we do and many physicians in many parts of the world do. We may not have to do it in every patient, but in patients with severe disease such as Ed, who had prednisone, who required prednisone, this is what he did. So this is what he used to do, and he did a clinical trial, and this is taken from the paper. He gave prednisone only to those patients who had eosinophils. The results were startling. Approximately half the patients showed no improvement, the other half had a dramatic improvement, and those who had an improvement were those patients who had eosinophils in their sputum. The local laboratory was useless, so he started looking at these cells himself. He found an old microscope from the attic, developed a rapid method which he shared with me just a few weeks before he passed away, insisted that I do that, and he said that my hands might get a bit dirty, but it would come off if I washed it well enough. Eosinophils are only present in the steroid responders. This was 1958, was completely ignored again until two pioneers in Hamilton, unfortunately, both of them passed away. Freddie Hargrave and his longtime friend and colleague, Jerry Dolowich, who was the head of allergy here, really wanted to implement this in clinical practice, not in research. And this is where the program, I was recruited to Hamilton to work with Freddie Hargrave in 1996. He had already worked out the method, simplified it to apply in clinical practice, and the task given to me as a postdoc was to evaluate its role in clinical practice. Now, just a word on it, it might sound simple, but it takes a very long time to validate a test. It took them 8 years of research, 110 research papers on just methodology before a test becomes good enough to be applied in routine clinical practice. He recruited postdocs who first showed that when you've got sputum, it's all yucky and goop and thick. You've got to disperse it so it flows easily. So the dispersion, you've got to evaluate the method that he used to disperse it. What reagent would be used to break up sputum? Would the reagent interfere with the cells in sputum? So you've got to get that done. And how do you get sputum in some people who absolutely refuse to spit? I've got the story. I'm Indian. So you saw in Paul's table, we are used to being at the bottom of most of those tables. But in this table about asthma, we are quite proud to be at the bottom. And we are very good spitters. You don't really need an induction. But there are some very conservative people who just absolutely refuse to spit. You've got to get them to spit. Hypertonic saline. So the method to get people to spit needed evaluation. Then how do you get smears? So how do you get a good quality smear? In order to stain, you've got to get a good quality smear. So that took a lot of research. Now when you spit, there's all sort of stuff in it. You've got saliva in it. So would saliva interfere with your cell counts? You've got to validate that, get the saliva out. So this, all I want to illustrate is in any test, before you apply it in practice, if you have rigor and methodology, the test becomes more reliable. And simple cell count in sputum took 8 years and almost 100 publications. The cytometry was validated. You've got to show that it's repeatable. If you do the same test twice, you've got to get the same results. If the patient gets worse, you've got to show that the cell's number goes up. If the patient gets better, you've got to show that the numbers come down, which is called responsiveness. So they did that, and then finally the test was there. It was all done. All I had to do was start applying it in practice. And that's how we do it. We collect sputum, we weigh it, we disperse it, we spin it, and then we stain it and look under the microscope. So essentially, just as you get a total and differential cell count in blood, we get a total and differential cell count in sputum. And if someone coughs up, if you've got an infection, you'll see one white cell called neutrophil. If you've got allergy, you see another white cell called the eosinophil, the pink cell. And we have looked at a large number of patients, and you can see a spectrum. Not all patients have got eosinophils. Some have eosinophils, some have neutrophils, some have both eosinophils and neutrophils. Others may not have any cells. It might all be the spasm. So really, depending on what we find in sputum, we customize treatment. And then what we showed is if we customize treatment based on what we have in sputum, it's much better. So what we did is we randomized patients with asthma into the standard strategy, which is one size fits all, or another strategy where we customize treatment based on what is in sputum. The benefits were significantly better. When we say customized sputum is if there was eosinophils, we would give more steroids. If there were neutrophils, we would not give steroids because we know that in most cases neutrophils would indicate an infection which would not require steroids. You would just treat with a bronchodilator for the spasm. We showed that if we customize, on average, we did significantly better. Now I'm going to move on to severe asthma and illustrate how if you apply biology to practice, we can really sort out even the most severe patients who require prednisone. Let's look at eosinophil. Eosinophil is a cell, a white cell produced in the bone marrow. All cells are produced in the bone marrow. They get into blood and then go to the organs and they cause their damage. Some very clever people had worked out that this cytokine, this protein, interleukin-5, is the key cytokine that is essential for these blood cells to be formed in the bone marrow. The gene is sequenced. We know exactly what cells produce it. It's expressed on eosinophils and the mature cells in the bone marrow. And a whole cascade of signaling mechanisms that this protein can trigger has all been worked out. So we know that this cytokine is a key cytokine that produces eosinophil. So it would be intuitive that if you can block the cytokine, eosinophils would disappear. And for whoever, if you believe in the theory of creation, and if you believe there is a creator, whoever the creator is, gave us eosinophils currently, as far as we know, only to kill worms. If you don't live with worms, we don't need the eosinophils. You can very easily get rid of the eosinophils and we'll be fine, as opposed to getting rid of the neutrophils and we'll be susceptible to infection. So if we can block these IL5, you should get rid of all eosinophils, which is what prednisone does too. But prednisone is like a cluster bomb. It's very effective, but there is collateral damage. whereas IL5 would be like your Tomahawk cruise missiles. Very, very expensive, rarely misses target, and wipes out that particular cell. So that was all fine and dandy. A company, GlaxoSmithKline, developed the drug, a monoclonal antibody to block interleukin-5, and it was evaluated in this trial from two centers in the United Kingdom and a center in Holland, and evaluated the effectiveness of this drug in an experimental model that Dr. Byrne highlighted, which is an allergen inhalation. Allergen causes eosinophils to go up in the lung, and if this is effective, it should block the eosinophils. And sure enough, it did. I won't go through this slide. It did block the blood and sputum eosinophils on the right panel. But the airway response that Dr. Byrne showed, the change in FEV1, wasn't blocked. So there was a lot of skepticism and cynicism in the scientific world that, well, this is just a flash in the pan. It blocks a cell, but it really doesn't make patients better. So the interest was dropped until Freddie Hargrave resuscitated the anti-IL5 program in Hamilton. It was around that time we saw an unfortunate 29-year-old woman who had very mild asthma for a long time, incredibly clever. She had been accepted into an MBA program at Harvard, had a baby, and then suddenly her asthma got worse. And she had significant sinus symptoms, wheezing, shortness of breath, colic, general malaise, and the eosinophils had gone completely berserk. They had come out of the bone marrow and gone to every organ in the body, not just to the lungs and to the sinuses, but to a gut, to a heart, to a skin, a condition associated with asthma called hyper-eosinophilic syndrome. We really had no treatment for her. This sort of New York skyline picture shows blood eosinophils on the y-axis on that side and the prednisone dose on the y-axis on this side, and all the treatment that we use. We use all sorts of nasty toxic drugs to try and get her off the prednisone, and nothing worked. She needed bucket loads of prednisone on the y-axis. We are on 15 to 20. She required up to 100 milligrams of prednisone every day, which is sufficient to make any sane person completely insane. Until we gave her this drug, mepolizumab, the anti-IL5 monoclonal antibody that had been condemned and put on a shelf, and it wiped out the eosinophils in this patient, and for the first time she was able to come off prednisone. So Freddie Hargrave hypothesized that the drug would only work in those patients whose asthma is truly driven by eosinophils. And I had shown you earlier that not all patients with asthma have got a lot of eosinophils in the airway. So if you have got severe asthma driven by eosinophils, we wipe out the eosinophils with a targeted treatment rather than with prednisone, they would all get better. In fact, that's exactly what we did. We hypothesized that, and we selected patients with severe asthma, those who were on prednisone like Ed, who had eosinophils in their sputum, by the method, stained by the method that Jerry Dolovich and Freddie Hargrave and his postdocs had developed, and randomized them to a clinical trial of either mepolizumab or placebo. And then we attempted to reduce the dose of prednisone, which had not been possible in those patients before. We gave 9 patients the active drug and 11 patients placebo. So this was a trial that was done in Hamilton with Dr. Byrne and Hargrave, and this was one of the first projects that was given to me, and to our satisfaction and the patient satisfaction, we were able to get these patients off their prednisone. Every patient was able to come off prednisone. Every patient who got the active drug came off prednisone, and every patient who got the dummy drug, the placebo, flared up, because we were trying to reduce prednisone by giving them a placebo, which is how the studies are done. So we really showed that the treatment worked, and now this has gone into large phase 2 trials, phase 3 trials, and it's likely to become approved in Canada and the United States very soon. Patients also felt better. This is another outcome called Asthma Quality of Life Questionnaire. Every patient who got the active drug felt better. Now, how do these cells come into blood? So they're produced in the bone marrow, and they spill over into blood, and they come into the lung. So we thought, well, in these severe patients, if we can block a cytokine, so they're produced in the bone marrow under the influence of this cytokine I talked about, interleukin-5, and another chemoattractant, a homing signal, CCR3, it's like the beacon that tells you where these cells, these are dumb cells, they don't know where to go. They're produced in the bone marrow, they just go out. Where do they go? So there must be some homing in signal, and that is facilitated by a chemokine called eotaxin that acts through a receptor called CCR3. So if we can block that, they would just stay in the bone marrow. So we thought we would do a second study to see if that would work. So what we did is we did a clinical trial where we gave for the first time a small molecule antagonist against CCR3 or placebo in those patients who had a lot of eosinophils, and showed that it didn't work. And that was a surprise. We just published this paper. So that's a bit of a surprise. Why would it not work? In these severe patients, we block the homing signal, it's still there, which would mean one of two things. Either a molecule wasn't good enough, or in these severe patients, perhaps the lung themselves may produce these eosinophils. They are not very dependent on the bone marrow producing it and spilling into blood. The so-called in situ eosinophilopoiesis. The lungs themselves perhaps could have the progenitor cell or some sort of early cell, a stem cell residing there that could turn into eosinophils without being recruited from blood. And there is a history of research on in situ eosinophilipoiesis in Hamilton. John Bienenstock, Judah Denberg, Dr. Byrne here, Dr. Mark Inman, my collaborator Roma Semi, and Gail Gofro. This hierarchy is not necessarily a sign of maturation or immaturation. It's just listed in a convenient way. That's all it is. It doesn't in any way take away, the maturity. But these investigators in their laboratories have shown that when you have an allergen exposure, if you have an infection, the cells are mobilized from the bone marrow, immature cells, and these immature cells, along with mature cells, get into the blood and then track into the lung. So there is a possibility that these immature cells could come into the lung and stay there. So this is a cartoon to illustrate that. That's the nose, pretty little nose, and then you've got the lungs there, and you've got an allergen stimulation. All sorts of chemicals are produced from the lung, which would then trigger the bone marrow to produce the progenitor cells and the mature cells. There could be maturation occurring in the bone marrow, and both cells would then track their way into the lung, and these progenitor cells could just stay in the lung. So we actually looked at that. This is a method that Roma Semi and Paula Byrne and Gail Gofro and Judah Denberg had developed. Immature cells are characterized by a particular characteristic, which is expression of a cluster designation antigen called CD34. So we collected sputum in our severe patients, enumerated the number of CD34 cells by flow cytometry. This is just a scatter plot. If you're familiar with flow cytometry, we use a laser to then look at the scatter of cells that float across the laser. And on the panel on the left here, you've got the number of those CD34 cells that produce interleukin-5. So our hypothesis is that there's a lot of these cells already in sputum, and therefore they would be able to produce IL5, and you don't need to recruit from blood. But the numbers are very small, but more than in patients or mild patients. Only about 0.61% of cells are immature cells producing IL5, and only 1% of cells, immature cells, 2% producing another key cytokine called interleukin-13. So it's really probably not the immature cell. But when the immature cells are stimulated with another cytokine that is produced from the lining of the lung in response to an allergen or a virus, IL33, they are able to produce IL5. So it's possible, but still the numbers are small, only 0.5% and about 4% on their top panel. So it's really perhaps not these immature cells in these patients with severe asthma. I won't spend too much time, we're running out of time, but there's another very important immunological cell which is called the ILC, innate lymphoid cells, which have been recently described. They are neither the classic allergy type lymphocytes or the infection type lymphocytes. We'll skip this for want of time, but the point I want to highlight is there is this population of cells which are really a bit of an orphan cell, which are able to produce these cytokines. And what we found very interestingly is in these patients with severe asthma who have got eosinophils who are not so much dependent on recruitment from blood, there is large population, 6% and 25% of these cells respectively producing IL5 and IL13 in the sputum. compared to 32 and 14% in blood. So there are these cells that are neither the classic immunological cells nor the progenitor cells that are able to produce cells. And therefore, if we can target these cells, rather than just looking at killing off the IL5, but kill the cells that produce IL5, it might be even better. So you see where I'm going. In patients with severe asthma, it's not probably the IL5, but the cells that are capable of producing IL5 that may have the receptor, it might be even better. And just as we were working on this trial was published, another molecule, a monoclonal antibody, benralizumab, that does not mop up the interleukin-5, but kills the cells that express the interleukin-5 receptor by a process called antibody-dependent cytotoxicity, ADCC. So it not only blocks IL5, but it kills the cell. So we thought this would be the appropriate way to treat these severe patients who require daily prednisone, who have got lots of cells, funny cells in the airway that produce IL5, and we are just about to embark on a clinical trial using this molecule in our severe patients. So far, fine and dandy. How do we identify these patients who have got eosinophils? We talked about doing cell count, but cell count is difficult. If you have a cough this winter, if you could just dip something into sputum and it would tell you if you've got eosinophils or neutrophils, that would be much better, rather than going to a laboratory and doing a cell count. So Freddie Hargrave developed a little kit to simplify processing sputum, and what we then did, we developed an ELISA to measure a specific eosinophil protein called eosinophil peroxidase. This, in collaboration with Jamie Lee at the Mayo, he identified a monoclonal antibody that would only pick up eosinophil peroxidase, which is only produced from the eosinophil. We validated this in sputum. So we took healthy controls, patients with a lot of neutrophils and patients with a lot of eosinophils, and this protein, EPX, was only detected in those patients with a lot of eosinophils. What we did is, instead of processing it, we started measuring it in unprocessed sputum, and we got an excellent correlation between just filtering the sputum and the processing after spinning it. So we could really, in principle, get sputum, filter it, and then measure it by a high throughput method. But it's not quite as dipping in paper. So in collaboration with John Brennan in chemical engineering, who is a wizard in putting anything on paper, we are in the process currently of putting this protein onto paper, a bioactive paper where you could dip your sputum and it would tell you exactly how many eosinophilic proteins are there and what would you do if you've got an exacerbation. So technically, if you've got a flare-up and if you do not get better with your bronchodilator, you dip it. If you've got more eosinophils, you increase the dose of steroids. And if steroids do not work, you could potentially use one of the monoclonal antibodies that would block the eosinophils. So essentially what I've discussed is to try and illustrate how, if you have a clinical medicine, if you can combine good clinical acumen, observation, skills and compassion, which are absolutely essential for the practice of medicine, with some creative thinking and basic biology, you could really be a better physician. Research training is not just to run a lab. You could apply it to patient care, and if you go through the full extent of translational medicine, you can make a huge difference to patient care. And I hope I've highlighted that personalized medicine, before it became a buzzword, was being practiced in Hamilton by Freddie Hargrave and Jerry Dolovich for almost 20 years, because they gave the right treatment to the right patient at the right time. And patient-centered research, which has now become a sort of buzzword in all sorts of granting application in severe asthma, which I've highlighted with the examples. One example will hopefully lead to better treatment for severe asthma. I love this pink cell, and if you're interested in things that are pink, I'd really like you to read this book, Eosinophils in Health and Disease, which will tell you everything you need to know about eosinophils. And there is this, we have got a secret cult society, International Eosinophil Society. We do not tolerate people who like any other cell. That's the only other problem. Thank you.